Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is April 29th, 2022, and we're talking to Hojoon Lee.
Yeah.
Did I get that right?
Yes, that's correct.
Good. Hojoon is an assistant professor in the Department of Neurobiology at Northwestern University. His lab studies taste, the taste receptors, molecular mechanisms of taste transduction, formation of appropriate connections in the taste pathway, and the central pathways that are responsible for taste-driven behavior. Hi, thanks for joining us.
Hi, thanks for having me.
With us today is Lindsay McPherson, UTSA's own taste specialist, and longtime collaborator of Hojoon. Hi, Lindsay.
Hi, Charlie. Good to be here.
And me, I'm your host, Charlie Wilson. So, Hojoon, the taste pathway is unusual in that the receptors are rapidly turning over, but the nerve fibers that carry signals from the receptors to the brain are not. And that means that the taste pathway is continuously facing a problem that most brain pathways face only once during development. That's a cool thing, I think, to study.
It is, yes.
The fibers have to reestablish their appropriate connections with receptors over and over again. So could you start with just a little summary of why this is necessary and what we know about how it works?
Yeah, so because taste information is absolutely necessary for the survival of the animal, it's important to maintain the fidelity of that information so that good nutrients such as sweet, which signals presence of carbohydrates, umami, which signals presence of amino acids or proteins, or salty, which signals essential salts, those would provoke appetitive and attractive behavior as opposed to bad things in potential food items such as sourness of a unripe fruit or bitterness of toxins, which have to be rejected. So in spite of taste receptor cells having to cycle and they only live about 2 weeks, so they have to cycle and continuously make new taste receptor cells to replace the old ones.
Why do they have to do that? Why can't they just live a long time like other cells?
I think it's because they're exposed to a lot of harmful things. So toxins might touch the taste receptor cells, which might kill them off, or you could bite on your tongue and lose some taste receptor cells, or burn your tongue and lose taste receptor cells. So you would want to maintain your sense of taste in spite of all these harmful injuries.
So the turnover is proactive and anticipation of me biting my tongue or eating something that's going to burn my taste receptors.
At least I think that's one of the advantages of turning over.
And then the taste receptor cells themselves are highly specific for a particular taste. So that means that if you lost all the taste receptor cells that were salty, you just wouldn't be able to taste salty anymore.
And not get the good salts in our diet. Yes.
And so that means the axons that innervate those also have to be completely specific, because this is a completely parallel, independent. It's another very odd thing about the pathway is it seems more completely parallel in independent channels that don't crosstalk very much.
Absolutely, and I think at the very base level, you don't get crosstalk between the good things versus the bad things. So you don't ever see sweetness and bitterness overlapping within the same pathway.
I could see the advantage of that, but it's also a problem for this turnover, because that means that a fiber that was getting input from some receptor cell, and then that receptor cell dies, and now that fiber, what's it going to do?
Yeah. Well, there are plenty of taste receptor cells to go around, and hopefully you'll be making enough new taste receptor cells to replace the ones that you have lost, and that would maintain the sense of taste. And this doesn't always happen, so I think there are many diseases where you lose the sense of taste. It could be transient or chronic, or maybe a more common story is we know of family members whose sense of taste has deteriorated and they don't perhaps make as cook as well as before, because now everything that your grandmother makes is saltier than it used to be. So with age, we do lose that sense of taste, partially because we don't replenish taste receptor cells as efficiently as before, or there might be other more central processing issues.
So what's the mechanism that keeps these pathways from getting confused as the receptors are turning over?
So when I first started working with Lindsay, we were interested in this question of how new taste receptor cells that are born would attract the correct fibers to make the connections correctly. And in order to make the correct connections, we thought that the taste receptor cells, or a newborn taste receptor cell, would produce signaling molecules that would attract the partner fibers from their partners to come and make the connections. And we did this by comparing expression of transcripts within bitter taste receptor cells versus those that are made in the sweet taste receptor cells. And we focused in on a signaling molecule or family of signaling molecules called Semaphores, because we saw enrichment of SEMA3A in the bitter taste receptor cell versus SEMA7A in the sweet taste receptor cell.
So an experiment like that, you're looking at sort of zillions of transcripts and searching through them for the ones, or you already have an idea about what kind of transcript you're looking for?
We had some ideas. We didn't know what we were looking for, really. But when we, tracked the most highly expressed or differentially expressed genes in the sweet versus the bitter population, we knew we were going to look for cell adhesion type molecules or axon guidance molecules, those types of things that might actually, do something for the fiber. And so once we looked at the top candidates, the Semaphores really popped out. And because there were these different versions of the Semaphore, and SEMA3A versus the Semaphore 7A in the bitter versus the sweet population, that was kind of a great happenstance that we saw that and we're like, okay, wouldn't that be cool if those two were really doing something to specify the connections between the taste receptor cell and the nerve fiber. And that's when Hojoon had really the great idea of manipulating the expression of those Semaphores within different taste populations. And so as soon as we saw those, and I remember we tested the first knockout of Semaphore 7A on our gustometer setup, and neither of us could believe it, but this mouse just was drinking quinine like nobody's business. Like, it was just going for it. And we'd never seen that in a mouse ever before, except for, maybe a total receptor knockout. So we knew we had something in our hands. So as soon as we saw that kind of behavioral phenotype in the mouse, Hojoon got onto the idea of doing these Semaphore swaps and misexpression profiles. And that really led to the confirmation that these were directing the connectivity, not just being permissive signals.
And for the students in the audience, I'd like to point out that it's important to maintain friendships and interactions with people in other labs, because we were very fortunate to be neighbors with the lab of Tom Jessel, and I had many friends, postdocs in that lab. So after seeing Semaphores pop up in our list, we raced to the lab and got hold of people who were working with Semaphores and asked them for the mice. So we were able to do the behavior experiments very soon after finding our top candidates, and that really expedited our research.
So in that mouse, bitter tastes like everything.
So this is one of the interesting things, because when we first saw Semaphore 3A, and we knew a little, when we looked up, a little bit about its history, and it's mostly known as the axon guidance repulsive cue. So we thought that potentially that the expression of Semaphore 3A would prevent the other taste neurons from attaching on to the bitter cells and receiving bitter information incorrectly. So that was our initial hypothesis. And we thought that might have still been the case when we had seen the behavioral deficit where the mice would keep drinking bitter. Was like, okay, well, maybe they're just messed up, right? And they just don't receive as much bitter input. But then when we went to look at the actual activity of the ganglion neurons, that is when our idea kind of changed, when our hypothesis changed. And because as soon as I went up to look at the activity of the ganglion neurons, I was expecting maybe that we would see no bitter responses within the ganglion, because we thought we had lost or really reduced the bitter sensitivity. Quite to the contrary. We saw all of these cells responding to bitter in addition to something else. So I was like, I remember coming right back downstairs. My imaging rig was upstairs. I came right back down to Hojoon. I'm like, well, it's definitely not a bitter knockout. Like, we see tons of bitter responses. And then we're scratching our heads, and we had to kind of reform our hypothesis to make more sense of this. And that's how we eventually identified that they're still getting to the bitters, but then they're reaching out and receiving inputs from the inappropriate cell types. So we would see bitter plus sweet, bitter plus sour, bitter plus umami, and all of these other combinations of multiply tuned.
So anything bitter would taste like a well-balanced meal.
Possibly. And then we did some experiments with this and trying bitter and sweet and getting it. Yeah.
But something won because the animals were willing to taste. They weren't repulsed by any of the bitter stuff.
And if we were to present mixtures, so bitter sweet mixtures, bitter typically wins out, and the animals were rejected. So there has to be some kind of a deficit in bitter sensation, or enough of a good thing to overtake that.
So for the bitter and sweet, you've identified the molecule, at least a molecule that's required in order for the axons to find their appropriate receptor. Yeah. Presumably there are more of those things to be discovered.
There are semaphores that are enriched in other taste receptor cells. Although we haven't been following up too much on it, we think that there are similar mechanisms working in the other systems too.
Is it a safe assumption that they'll be similar? There's so many things that aren't similar
that's true.
Between receptor cells.
Right. And of course, there are other axon guidance molecules that are enriched in these separate populations. So the contributions of these connectivity molecules in wiring also has to be explored.
So the ganglion cells' projections into the brain also stay pretty separate in the next place. That's the NTS?
Yes,
that's the nucleus of the solitary tract.
That's correct.
So there also cells have a specificity for, is that true?
There's a paper that came out last year in Cell by Jin et al., where they describe molecular markers for the individual population. So they've at least found that the sour goes through prodynorphin expressing neurons. Bitter taste goes through somatostatin expressing neurons, and then sweet goes through CalB2 expressing neurons. So, and they've been able to fool animals into thinking by stimulating these neurons that they would elicit some kind of taste-dependent behavior.
So maybe the same molecules or some similar molecules are controlling it all the way through? I mean, is that
so we had the hypothesis that because the ganglion neurons are pseudo-unipolar neurons, so they have their cell bodies in the ganglia, in the ear, and then they would send out their axon, and the axon splits. One arm goes to the tongue, and the other arm goes to the brainstem in the NTS. So we thought perhaps if this arm that goes to the tongue requires semaphorin for wiring, that perhaps the brainstem NTS neurons express SEMA3A also, and that's how they establish this connectivity. And although we haven't really followed up on it, we have seen expression of SEMA3A discretely in the NTS, so perhaps that model is still valid. Just haven't tried hard enough to look at it.
Interesting thing about the NTS though too is that's like the first place that you can have, or at least we think this is like the first relay station that's not completely just a relay. I mean, there's some computations and signaling that happens within that nucleus that we don't believe really happens at all or much at all in the ganglion itself. So the NTS is, I think, one of the kind of the next steps to figure out where all this taste information is going, and then also where some of this feed forward and feedback circuits integrate.
This NTS is a central hub of information where there's input from the vagal system, so visceral information, and some of the internal state information. So there has to be some integration of all these different information to regulate taste-dependent behavior or ingestive behavior and vice versa.
So it's still a ways. I mean, you show these children tasting something sweet for the first time and reacting to that. It's still a ways from NTS to those reactions. What do you envision? I mean, is it your goal to track down the whole thing from the taste receptor to the motor output?
Some of it. I think we would like to understand how the taste system regulates some of the autonomic nervous system, the parasympathetic nervous system, to achieve some of these motor output. So at the moment, we're interested in looking at salivation and how tasting something sour might induce salivation, or gag reflexes, how bitterness might make you gag. And those are more local brainstem circuits, which I think is the first step towards understanding how we do more complex reactions and behaviors.
Yes, because some of those kind of more basal level reflex circuits don't require the cortex. You can take the cortex out of a mouse and still get it to gape to bitter.
Yeah.
So those will be probably the first things that we can tackle, and then it gets more complicated from there.
Yeah, but that's quite a bit already. So that the reactions are different enough, maybe, that the reactions are tastes. Since it, like salivation is, I mean, everything can cause salivation, but salivation is especially connected to sour, right?
Yep.
So if you were following the neurons that respond to sour, you might follow them right away into neurons that control salivation.
Yeah, that's our hope. And I think it's an exciting time to be a neuroscientist with all these new tools that have come up in the past couple of years. And we are able to record neuronal activity at a larger scale with much more ease than before. We're able to manipulate select neurons, activate them, inhibit them. And I think that really gives us a lot of power and tools to look at circuits and how circuits are established. So those are some of the tools that we're trying to incorporate in our studies.
You mentioned gagging. I mean, what causes gagging? What taste could cause gagging?
If you put bitter toward the back of your tongue in the mouth, it will elicit this thing. It's similar to a gag, but we call it a gape reflex, and it's just opening the mouth and kind of like that.
And that's specific to bitter.
It's pretty specific to bitter. You can't elicit it in addition to other stimuli that you've paired with something bad. So you can't elicit a gag with like a conditioned taste or conditioned flavor avoidance, aversion. So that can elicit a gag, but that's more top top down. So, but you can get one directly without any cortical input, just by putting a bitter substance in the back of the tongue.
So that's what do we know about what causes that response? I mean, that if that's specific, that would be pretty close. That's not a long way away from the neurons you're recording from. Do you think?
No. Yeah. So I'm really interested in it. we think as we're working on more taste reflex in the back of the tongue, which incorporates a different ganglion than the innervation in the front of the tongue. It's this petrosal ganglion that's fused to the vagal complex, known as petrosal jugular complex. And potentially those are the ones that are more responsible for eliciting these kind of disgust, gape type reactions. So it may be possible to, figure out a way to tease these apart by comparing these circuits from front of the tongue neurons versus the back of the tongue neurons. So we're definitely interested in doing this.
So what's the difference in the receptors between the front and the front of the tongue is specific for good things and the back for bad things, or is it?
No.
Not?
So we have the receptors for all of them in all the taste buds, or the vast majority of taste buds. There's a slight difference in the proportions of cells, very slight, but they're pretty much all there. The only thing that really is different between the front and the back is salt taste, at least in the mice. There's only salt taste receptors present in the front. There's very few, if any, in the back. But yeah, so the receptors themselves and the taste receptor cells are pretty, by and large, fairly identical between the front and the back. But we think the meaning of activating those receptors is different depending on where.
Just by virtue of where it is.
Yes.
Because you're not just sampling it anymore.
Right. By the time it hits the circumvallate papillometer way back in the tongue, they're pretty much close to being ingested. It's like right before you hit the esophagus. So at that point, you have decided that you're gonna swallow this thing. Maybe it's a bad idea, and maybe this is the last chance you have to gape and spit it out.
Huh. But these are, are there taste receptors on the palate?
Yes.
And do you know anything about them? I mean, I don't use this kind of language, but I hear people talking about taste and the palate as if the palate is the most sensitive part of the taste system or something. Is that not right?
I think it's just a shortcut for saying there's two olfactory pathways too. So there's the orthonasal and retronasal pathways of olfaction, and they're different too, right? So there's the retronasal pathway is when the food is in your mouth and you're kind of smelling it from behind your palate, which hits the olfactory epithelium. And that is more like when we have a palate, if you're tasting wine or something, you sense it on your palate. That's more the retronasal olfactory system combining with taste.
We are so confused about what we smell versus what we taste.
For sure, because they happen at the same time. They're almost like, unless you have a really severe case of a cold or COVID, potentially, and your olfactory system is wiped out, or you've zinked your nose by accident or on purpose, taste is extremely limited. And so what we experience as as taste is flavor, which is a combination of taste plus smell.
And for the taste researchers, our definition of taste is very, very precise. So we typically have been influenced by scientists who used to take out the taste nerve, especially the chorda tympani, and they would hook up an electrode to measure whole nerve activity. So anything that goes through that, anything that elicits activity in the chorda tympani nerve, we define as taste. So those would be the 5 basic tastes, but that eliminates capsaicin, which is why spiciness to us is not a taste.
But in your experiments with mice, how do you avoid olfactory stimulation and confusing the mouse between those?
It's a toughie, and it's not pleasant. We had to do this for some of our experiments with the bitter taste.
You have to get rid of the olfactory.
Yeah, Hojoon did these experiments. He pulled out the olfactory bulbs and
Thanks for calling me out.
Yeah, sorry, Hojoon, sorry.
So there are chemical methods for removing the olfactory epithelium, but they're not as reliable as removing the olfactory bulb. So we've actually had to go in and remove the olfactory bulb, because what we were seeing is that when we do behavioral experiments with bitter tastants, the animals sometimes would recognize the bitter taste from the smell without even having to turn around. So they would not even look at the bitter spouts that's presented. So we figured out that this is a problem. We can't determine bitter taste sensitivity if they're already recognizing it without putting their tongue on it. So that's why we resorted to removing the olfactory bulb. And that really helped us understand the taste system better.
So why do we have both? I mean, did olfactory come later and animals had to get along with just taste at some point, or?
I do think that taste is more primeval, because you have to have contact with whatever the substance is, and like, I don't know, we all came from the ocean.
So a big difference is that the taste system relies on detectors, or these receptor cells that are epithelial derived. So this likely evolved prior to evolution of neurons, sensory neurons, whereas the olfactory sensory neurons they're neurons. So I like to tell my olfactory researching friends that the taste system came before the olfactory system. It might have preceded the development of synapses, development of sensory neurons, because it's that important that you need primal animals needed to detect the presence of nutrients to survive.
And it doesn't use synapses. The taste receptors somehow can communicate with the nerve fibers without making synapses.
That's correct. Most of the taste receptor cells, namely the sweet, umami, and bitter, they don't form prototypical synapses, and they're able to communicate by squirting out ATP to the afferent fibers that supposedly are close by and still able to maintain that fidelity of information.
What do you mean supposedly are close by?
I mean, they don't form synapses. So people have made electron micrographs, and they don't make synapses as we recognize them. So it's still a mystery on how they can efficiently signal to their partner fibers. And this is something that is not very common in our body. It's what might be present in Merkel cells and other systems where there's this kind of ephaptic coupling. So they come in close contact but not make actual synapses.
But it is chemical transmission.
It is chemical transmission.
We talked to Tom Finger just a few weeks ago about the receptors.
Yeah.
The reason for synapses, I was told as a student, is to maintain private communication. So if we were just to release our transmitter into the extracellular space, who knows who's going to hear it? But if there's a nice synapse, then it's just going to one thing.
Which always intrigued me about the taste system, because is it private or is it just public information, right? Yeah. Once the taste receptor cell is activated and ATP is secreted, the fiber is just there, and if it has P2X receptors, which we know it does, it will become activated. How does that information become private so that not every fiber nearby gets activated? So I think what's coming more clear lately is this role of type 1 taste receptor cells. And these are glial-like, and they tend to ensheath around. They look like taste receptors, but they don't have taste receptor. They look like taste receptor cells, but they don't have taste receptors. Right. What they do have are ATP pumps, and they will suck up a bunch of ATP. And if they act likely, as I think more people are coming around to think of them as, that they help protect this contact between taste
So then they
receptor
create
cells.
a diffusion barrier, so the ATP can't diffuse just anywhere. It has to do. So that means that the fibers, I want to say postsynaptic fibers, but they're not postsynaptic.
Yeah.
They're just
Well, kind of.
fibers. Those fibers must be in some kind of spatial relationship to those type 2?
Type 1.
Type 1 cells.
Yes.
And that we could look at it and see that the diffusion pathways that the cell would be left with.
That definitely is the general idea. I think also with the recent identification of this voltage-gated ATP channels called CalHMs, they seem to be positioned intracellularly. They have the mitochondria and the ATP pumps, ATP pumps closely positioned to where the CalHM channels are, and as they pump out ATP, there invariably seems to be some fibers, afferent fibers, positioned close by to take up those signals. So although we don't really understand how this organization happens at this point, and especially the molecular aspect of it, we are starting to see some glimmers of hope for understanding this system.
Great. That's a great ending to our story today. So thank you very much, Hojoon.
Thank you for having me.
For joining us, and Lindsay.
My pleasure.
And by the way, this concludes our podcast series for this semester. Neuroscientists Talk Shop will be taking a break for this summer, but our series will resume again in August. See you then. This has been Neuroscientists Talk Shop.
Thank you.